so these are going to be the extraocular eye muscles that move your eyeballs around. So the superior or the levator palpebrae superioris that opens the eye, and the orbicularis oculi that closes the eye are not technically part of those extrinsic eye muscles. They're more like muscles of the eyelid. These extrinsic eye muscles actually move the eyeball itself. And so we have 6 of them. 4 of them are rectus muscles and 2 of them are obliques. And their names are very descriptive. And so the medial rectus is going to be located on the medial side of the eye. Rectus means straight. So it's the muscle that goes straight back on the medial side of the eye. It pulls the eye medially. It's innervated by cranial nerve 3, the oculomotor nerve. The lateral rectus is located on the lateral side of the eye. It goes straight back and it pulls the eye laterally, and it's innervated by cranial nerve 6, the abducens. The inferior rectus is located on the inferior portion, and it pulls the eye inferiorly and a little bit medially. And it's innervated by cranial nerve 3, the oculomotor nerve. The superior rectus pulls the eye superior and medially, and it's located on the superior and goes straight back. It's also innervated by cranial nerve 3, the oculomotor. So all of these rectus muscles are innervated by cranial nerve 3, the oculomotor, except the lateral rectus, which is innervated by cranial nerve 6, the abducens. Now notice that all of these are also, the ones that are innervated by the oculomotor nerve, they all are also going to pull medially. And so all of these pull medially because when you are looking at things close, part of the accommodation reflex is for your eyes to converge. So you're going to be looking together. And so you're going to be more likely to be converging your eyes and moving your eyes medially. So that would be why this one nerve innervates all of these that are going to be pulling the eyes medially, and that most of your muscles are going to pull them medially versus only the lateral rectus pulls it laterally. So we do still need to pull our eyes laterally sometimes, but not as often. And it has the lateral rectus has a different nerve for that. Now I said there's 2 others called the obliques, and these oblique eye muscles, we have an inferior oblique, which is on the oblique. Instead of going straight back from the eye all the way back, they go at an angle. And so the inferior oblique elevates and turns the eye laterally. This one is also going to be innervated by cranial nerve 3. It innervates most of them. The superior oblique depresses the eye and turns the eye laterally. And so this one passes through the loop like a pulley-like loop called the trochlea, and it's innervated by cranial nerve 4, the trochlear nerve, right? It's trochlear nerve. So cranial nerve 4 and 6 are the 2 weird ones, and most of the others are innervated by cranial nerve 3. So we can see here the muscles that go straight back are going to be the rectus muscles, and then the muscles that kind of are twisty and weird are the oblique muscles. So here's our superior oblique and our inferior oblique. And then we can see the orbicularis oculi here, and then we would have the levator palpebrae superioris. We just can't see it right now. And this is the other view of it from the other side. Now, if we removed the eyeball, we could see how the muscles themselves actually run. So you can see how the rectus muscles go straight back, whereas the oblique muscles go to the side. So we have the superior oblique on the top and the inferior oblique on the bottom. We do have a model that has all of the eye muscles on it, so you'll be able to label those in the quiz. All right, and a couple of cool widgets where you can see the eyeballs moving around. So now the structure of the eyeball itself. The eye is almost spherical. It's about 2.5 centimeters in diameter, and it's located in the orbit, padded by this periorbital fat. It's kind of around the eye. It has 2 cavities within the inside of the eye, the posterior cavity, which is everything behind the lens, and the anterior cavity, which is everything in front of the lens. The anterior cavity is then subdivided into anterior and posterior chambers of the anterior cavity, and we'll get to that in a little bit. But we're going to talk about the outer wall first, and the outer wall is formed by 3 tunics. The outside tunic, or the fibrous tunic, the middle tunic, or the vascular tunic, right, it's full of blood, and the inner, or the retinal, or the neural tunic. And so we can see that this layer here shows the different tunics. So our fibrous tunic is on the outside. It's made of a lot of fibrous tissue, the sclera, and so this is going to give the eye a lot of strength. And we would have dissected sheep eyeballs in lab, and you could, as you poke the scalpel through, you can see that the eyes are pretty strong. That's going to be the sclera, giving it a lot of that toughness and keeping it in shape. And then we have the clear cornea on the outside. So the fibrous tunic is made of the white sclera and the clear cornea. That's all the outside. Next, we have the vascular tunic. The vascular tunic is going to be made up of 3 different pieces. So the iris is the colored portion of the eye. This is in front of the lens. It's within the anterior cavity. The iris is also going to have the pupil, which is the opening in the middle. Then we have the ciliary body right here. And the ciliary body is kind of like this part at the very edge of the iris, as well as it's these little lumpy things here. And these little suspensory ligaments that are connected to the lens are going to be connected there. And the ciliary body is made of two things. It's made of the ciliary muscles, which are going to connect to these suspensory ligaments and then change the shape of the lens for focusing at different distances. And then they also have the ciliary processes, which are going to be important for circulating this fluid through the front of the eye called the aqueous humor. Yep, we went through there. All right. So the iris, once again, this gives the eye the color. So the iris, I mentioned earlier that the anterior cavity is going to be subdivided into two chambers, an anterior chamber and a posterior chamber. So the iris is going to be the dividing line in between the anterior chamber and the posterior chamber. So all of this is in front of the lens. So the anterior chamber is going to be between the cornea and the iris, so the very front, and the posterior chamber is between the iris and the lens. And so really the anterior chamber is what you can see, right? Because you can see from the cornea to the iris, and that's pretty much it, unless you're looking through with an ophthalmoscope or a slit lamp or something. And the pupil is the opening in the center of the iris that connects the two chambers. The iris controls your pupil diameter. It lets a certain amount of light into your eye, so this is pretty important. We have a couple of different muscles. So the sphincter pupillae muscles, these are circular fibers that constrict the pupil, and they are parasympathetically innervated by cranial nerve 3. The dilator pupillae muscle obviously dilates the pupil. These are radially organized, so sort of like the rays of a sun, they go out. And that smooth muscle is going to dilate the pupil with sympathetic nervous system activity. So those are kind of the two opposite effects. The pupillary light reflex alters the size of the pupil in response to light. So increased brightness leads to constriction of the pupil, so we don't get too much light back there. We can see a good picture of this here. In low light, the pupils dilate, and in bright light, the pupils constrict. Now the last layer is called actually before I do that, I want to mention this picture right here. So here is the posterior cavity, here is the anterior cavity, and we can see how the iris here is going to divide the anterior chamber up here from the posterior chamber, which is this small area right here. And then I'm going to show here, this is the retina. So the retina is going to be this last deepest layer or deepest tunic right here. And the retina is going to be also known as the neural tunic. And it's made of a couple of different layers. Let me get back to it. Here, the pigmented layer in the back, this is attached to the choroid, which, did I mention what the choroid does? No, I don't think I did. The choroid is going to be part of the vascular layer, and it's this back part all the way here. So we have the iris and the ciliary body, and then the choroid. The choroid is the whole back area that has all of these blood vessels in it, and it basically nourishes the retina. The retina sits on top of the choroid. All right, so now we'll get back to the retina, right? So the pigmented layer sits right on top of the choroid, and it provides vitamin A for the photoreceptors. And we'll get into the photoreceptors in lecture. It is also very pigmented, right? It's dark, so it absorbs stray light to prevent light scatter. Then next we have the neural layer, which is the layer that houses the photoreceptors and the neurons, so the rods and cones. It receives light and converts it into nerve signals. The ora serrata is kind of the jagged edge between the photosensitive in the back and the non-photosensitive in the front parts of the retina, right? The front of the eye doesn't really need to see things, just the back. So because the front of the eye doesn't get light. The non-photosensitive part is anterior, and it covers the ciliary body and the posterior side of the iris. So the retina is going to be made of nerves. It's made of 3 different layers, and they're mostly made of neurons. So the photoreceptor cell is the outermost neural layer. It contains rods and cones. These are special photoreceptors. They're special cells that contain pigments that react to light. The next layer is the bipolar cell layer. They are bipolar neurons, right? They have one axon and one dendrite. We said they were special. We only found them in the olfactory epithelium in the eye. The dendrites receive synaptic input from the rods and the cones, and then they send that input to the ganglion cell layer, which is the innermost neural layer. And their axons are going to gather at the optic disc and form the optic nerve. These are capable of action potentials just like the others. Now, there are a couple of interneurons between them, the horizontal and amacrine cells. We're not going to talk about them as much, but they kind of regulate the signals. that are sent between those cells. So here, this is going to be our neural layer up here. This is the choroid, nourishing it with lots of blood vessels. Then we have this pigmented layer here, that's a darker layer, and here's the photoreceptors. So these guys here are the cones, the blue one, and then we have the rods are going to be the yellow ones. Now this little pinkish layer is the bipolar cells, and then finally we have the ganglion cells. And all of the axons here are going to run together, and they're going to go to a part of the retina called the optic disc, or the blind spot. And that's where the optic nerve is going to be leaving from the back of the eye. So the light is going to pass through this way, but then the action potential is going to start in the photoreceptor cells and go this way and then out. And we're going to talk more about exactly how that happens in lecture. So now if we looked at the retina, the back of your eye, like with an ophthalmoscope, or you have a picture of the back of your eye from when you went to the eye doctor, it would look like this. So we're going to look sort of at the big area now, not really at the cells. So we have a couple of important structures. This right here is the optic disc. You can see it's where all the blood vessels are running through. And also, you can't really tell, but it's where all the axons of those ganglion cells are running to. And so they're all going to exit the back of the eye right here at the optic nerve, and the vessels are also going to exit there. So wherever the vessels are going, that's going to be the optic disc. And this is also the blind spot. Now here, if we look just lateral to it, we can see this like darker area here. This is going to be like the whole area is called the macula glutea, which is like the yellow spot. I'm not really sure. It doesn't look that yellowish to me in this picture, but it's called the macula glutea, and it has like a little pit or depression at the back called the fovea centralis, right? Fovea is for a pit or depression. This is where most of our cones are going to be developed. This is where most of our visual acuity is, and this is pretty much where anytime you're looking at something in color and in focus, that's where the light is going to be focused on. And this is also an area where if you have macular degeneration, that's the macula here. That's going to be degenerating. It's the problem with those cells. Let's see. I think that's about it. The peripheral retina, that's everything outside. So this part is the peripheral retina, is going to be mostly filled with rods. So the cones are going to be filled, concentrated at the fovea centralis, whereas the peripheral retina is going to have mostly rods, and it's mostly efficient in low light, but it's not quite as, rods do not see color, and they're not quite as sharp. So next, as we're moving through the eyes, we've kind of looked at all the different layers of the eyes, and now I want to talk about the lens. And we said the lens is the thing that is going to divide the anterior cavity from the posterior cavity. The lens is also important, though, because it can change shape, and it can focus the light on the retina. So this is made of cells, but it's actually very clear, and it looks kind of crystally. All these cells have lost their organelles and are filled with a crystalline protein. The lens is enclosed by a dense fibrous elastic capsule, and it can actually move. And so it should be able to bend light, and so it should be fairly clear. The shape of the lens determines how much the light is bent, and that's going to determine how it's focused on the back of your eye. The shape is determined by the ciliary muscle, right, which is part of that vascular tunic, and the suspensory ligaments. And those are the ligaments that attach the lens to the ciliary muscle. So when viewing objects that are far away, 20 or more feet away, the ciliary muscles relax. The suspensory ligaments then tense up, because when they relax, it kind of dilates the eye area, and it's going to relax, and it's going to flatten out the lens. It kind of pulls the lens flat when they relax. When you look at something close, the ciliary muscles are going to tense. The suspensory ligaments then are going to loosen, right, because we're constricting a little bit, and then the lens becomes more spherical, and it rounds up to undergo accommodation. We'll mention that again in lecture. But we can see here that the ciliary muscles are relaxed. It's going to pull the lens taut and flatten it, and then we can see things that are far away. If we're looking at things close up, we're going to contract these ciliary muscles, sort of constrict them like that. It's going to loosen up these suspensory ligaments, and it's going to round up the lens, and we can see things close. So cataracts are basically going to be opacities within the lens. So if the lens gets cloudy, and we can't really see as well because, right, light is supposed to be passing through that. So if it's cloudy, obviously we're not going to be able to see it as well. It needs to be removed. Any of these opacities can be removed when it interferes with normal activities. And so we can remove the whole lens and then replace it with a, let's say fake lens, but an artificial lens. So other things we'll talk about are going to be the vitreous humor and the aqueous humor. And these are the like fluid and jelly-like things that fill up the eyeball. So the vitreous humor fills up the posterior cavity of the eye, which is behind the lens, and it sits and pushes back on the retina. It's transparent, it's gelatinous, and it was first produced in embryonic development, and it doesn't get refreshed. It's always kind of the same. It helps the eye maintain the shape. It keeps pushing the retina back against the choroid. And it is sometimes, if you get these jelly-like pieces of the vitreous humor that kind of break by and sort of like float around in the back of your eye, that is what you see when you see floaters. If anyone, like if you look at something really bright and you see little things floating across your eye, it can be parts of that vitreous humor. The aqueous humor is the fluid in the front part of your eye. This is more like the CSF in that it's constantly produced and constantly reabsorbed. And so this is transparent and watery in the anterior cavity, so in front of the lens. So it's going to be bathing the inside of the cornea as well as surrounding the iris. And it's produced by the ciliary processes, and it nourishes and oxygenates the lens in the inner cornea. And so we'll talk a little bit about how it circulates and how it drains. So the plasma from the blood is filtered across the capillary walls within the ciliary processes, kind of like it happened in the choroid plexus for the CSF. The aqueous humor, so we're going to like push the water out. It's going to become aqueous humor. It's going to go into the posterior chamber. Then it's going to circulate through the pupil and go into the anterior chamber. And it's going to drain there via the scleral venous sinuses, just like our CSF eventually drained in the dural venous sinuses. If we produce more, but we don't drain enough, it's going to start filling up the eye and increasing the pressure inside the eye, and we call that glaucoma. So this picture here is going to kind of show these ciliary processes are going to be in here. So the aqueous humor is going to be created and excreted into the posterior chamber here, and it's going to go up through the lens into the anterior chamber. It's going to kind of circulate and nourish things, and then it's going to go and be recycled at the scleral venous sinus. All right, so that is it. The two anatomy zone videos are really good, so I'd watch those. And if you have any questions, let me know.